Hi everyone, thank you for joining my talk. My name is Ming and I am a second-year master's student at the Friedrich Alexander University of Erlangen-Nuremberg in Germany. I will be talking about the effect of sampling on extinction selectivity and the simulation-based approaches we take to understand these uncertainties. Extinction selectivity studies seek to understand why, in the light of crisis, certain taxa go extinct while others survive. The quantification can help us understand diversity metrics during background and mass extinction intervals and predict which taxa are most at risk today. Extinction selectivity is generally measured on ecological and physiological traits, such as whether a taxon in an ecosystem is benthic or epibenthic, has a wide geographic range or a small geographic range, or is small-bodied or large-bodied. In my project, I will be assessing body size and extinction risk. One method of assessing selectivity is by applying logistic regression methods in time slices. Each taxon in a time bin either survives, coded as 0, or goes extinct, coded as 1. The log odds ratio measures the strength of the association between the variable and the outcome. Log odds significantly greater than 0 indicate a positive association, and log odds close to 0 indicate no associations. Therefore, in this 2016 study assessing ecological traits and extinction risk, positive size coefficients indicate that larger taxa are at risk, while negative coefficients identify when smaller taxa are more at risk. The study concludes that small taxa were preferentially at risk during ancient mass extinctions, while in the modern oceans, uniquely, large taxa are more at risk of extinction. We know from Phanerozoic trends in body size that a Lilliput effect is expressed in animal lineages. That is, there is a pronounced reduction in body size in the aftermath of mass extinctions. These could be due to small taxa surviving, changes in relative abundance, repopulation by small taxa, and dwarfing of larger fauna. In this current study, we hypothesize that small taxa are more likely to survive mass extinctions. By preferential extinction of larger taxa, the average body size decreases to produce the Lilliput effect. In a 2020 reassessment of body size and extinction risk by capture-mark-recapture methods, Payne and Heim concluded that in the Phanerozoic, smaller body taxa are more likely to go extinct, as indicated by the negative coefficients on panel A. In addition, small taxa are also less likely to be sampled, indicated by negative coefficients on panel B. However, size selectivity of extinction here is contrary to our understanding of the Lilliput effect. We hypothesize that sampling completeness may account for this discrepancy. We have famously heard that the fossil record is incomplete. A quantification of all Phanerozoic marine invertebrate fossils using occurrence-based data from the Paleobiology database shows us that many intervals are only 50% complete. This is important because a current limitation of extinction selectivity is that we take recorded stratigraphic ranges at face value. However, the Singer-Lipps effect tells us that the youngest known fossil occurrence is not the actual last occurrence date and is only an approximation. As demonstrated in this figure, a simultaneous extinction event looks quite different if only the outcrop that is highlighted in yellow is sampled. The backward smearing of last appearance dates causes the extinction event to appear gradual. We cannot, therefore, only take stratigraphic ranges recorded in the Paleobiology database at face value. As we don't know the effect of sampling on usual methods of selectivity, we use a simulation-based approach in R. We first simulate life and biodiversity, and then we control the fossil record by introducing gaps in the data. We then apply diversity dynamics metrics using the R package divdin. Finally, we assess selectivity using logistic regression methods in R. To simulate our control records, we generate stratigraphic ranges of thousands of taxa. We then introduce an extinction event. In this figure, the first 3 taxa go extinct at the boundary, while the starfish survives, so we truncate our stratigraphic ranges. After the extinction event, we have originations of new taxa, and in this schematic, 2 more mollusks originate. We repeat this process 5 times to create a complete Phanerozoic record. In the first complete scenario, extinctions are random. In the second record, bigger taxa preferentially go extinct, and in the third, smaller taxa preferentially go extinct. All three of our complete records are then degraded by removing occurrences from our data set. When we randomly sample our data, we collect big and small taxa evenly. The complete record is marked in red. When we collect 80% of our fossils, our recovered dataset becomes smaller. We progressively degrade the record, but the samples are all random and not biased by size. This process is repeated for all three of our extinction scenarios. We also introduce size bias sampling when we decrease our fossil records unevenly. As demonstrated by the cartoon, the complete record includes 10 small taxa and 10 large taxa. When we weight our sampling, we collect 80% of the shells and 20% of the sharks, 70% of the small taxa and 30% of the big, or more evenly with 60% of the shells and 40% of the sharks. This cartoon demonstrates sampling bias when smaller taxa are better sampled. We also repeat this process to collect datasets where bigger taxa are better sampled. Finally, we use logistic regression methods to analyze the association between body size and extinction risk. In this slide, our extinctions are random, and we did not select for size. The sampling is also even, that is, the probability of collecting small fossils is the same as the probability of collecting large fossils. As a reminder, positive coefficients indicate bigger taxa are more at risk, and a negative association shows that smaller taxa are more at risk. The complete record, marked in dark red, shows that there is a weak association between size and risk. However, in the Middle Cretaceous, there is one instance where there is a significant risk for bigger taxa. Degrading the fossil record to have 80%, 60%, 40%, and 20% sampling does not significantly alter our selectivity results. Things become a little more interesting when we have a simulated fossil record where bigger taxa are preferentially going extinct. In this slide, small taxa have a higher probability of being collected by paleontologists. The complete record there shows that there are two instances where the p-value is significant. In the late Cretaceous, bigger taxa are more at risk. When we start degrading the record by sampling small taxa much better than larger, we start to notice many more instances where coefficients indicate significant extinctions for large taxa. The difference between the complete record and the degraded record are most obvious when we look at the subset containing 85% of small taxa and 15% of large taxa. In the Triassic, extinction risk in the complete red record shows instances where big taxa are at some risk, but they are not significant. However, regression coefficients from the degraded, gray record identifies 3 significant intervals where big taxa are very much at risk of extinction. In the Jurassic, we observe this again. In fact, we also see instances where the complete record indicates smaller taxa being more at risk, but the degraded record suggests otherwise. In the complete Cretaceous record, there are only 2 instances where big taxa are significantly at risk of extinction. However, as our degraded record samples big taxa very poorly, coefficients suggest that big taxa are significantly at risk many more times. In the Cenozoic, our simulated completed record suggests no associations between size and risk, but our degraded record identifies multiple instances where smaller taxa are significantly at risk of extinction. We observe the opposite when we have a simulated fossil record where big taxa preferentially go extinct, but big taxa are more likely to be collected by paleontologists. The complete record shows that there are 2 instances where p-values are significant. However, the degraded record shows that there are many more instances of taxa being at risk. Results are clearest when we look at the most uneven record. That is, 85% of the collected fossils are big and only 15% are small. In the Triassic, both body sizes are at risk of extinction, but not significantly so. However, because our small-bodied fossils are sampled so little, there are 3 instances of small taxa being significantly at risk of extinction. In the Jurassic and the Cretaceous, the complete record does not indicate significant risk of extinction due to body size. However, throughout the two periods, small taxa appeared to be more vulnerable in our poorly sampled data. In fact, there are two instances where the complete record does indicate preferential extinction of big taxa. However, the degraded record shows us the opposite result. In the Paleogene and Neogene, even though big taxa are better sampled, they appear to be more at risk than suggested by the full record. These bring us to our conclusions. Our simulated extinction scenarios are highly sensitive to the quality of sampling. When bigger taxa are better sampled, small taxa appear to be more vulnerable to extinction, whereas when smaller taxa are better sampled, big taxa appear to be more vulnerable to extinction. This suggests a selectivity of sampling rather than a selectivity of size. To circle back to these results about the association between extinction and sampling and body size, we can therefore say that we need to take sampling and size as covariants. We therefore propose that capture mark recapture models be used, which can take sampling and size and other factors as covariants. A lot of people were involved in the process and in this project, and so here is a shout out to my co-authors. Thank you for your time, and I'm happy to answer any questions.